Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to be talking about cancer and apoptosis. So cancer arises when the normal regulatory mechanisms that control the cell cycle are disrupted. This leads to uncontrolled cell proliferation. In healthy cells, checkpoints and regulatory proteins like p53 and Rb ensure that DNA is undamaged and fully replicated before the cell divides. When these safeguards fail, cells may continue dividing despite mutations, damage, or lack of proper signals. These are the hallmarks of cancer. So one of the most important tumor suppressors is p53. This is often referred to as the guardian of the genome. It is activated in response to DNA damage, and it can pause the cell cycle at the G1S checkpoint by inducing the expression of p21, which is a CDK inhibitor. Now this gives the cell time to repair its DNA before replication. If damage is too extensive, p53 can initiate apoptosis. Now mutations that inactivate TP53, which is the gene encoding p53, remove this checkpoint. This allows damaged cells to continue dividing. Another key tumor suppressor is the retinoblastoma protein, or Rb. This controls the G1S transition. In its active, hypophosphorylated form, Rb can bind to E2F transcription factors and prevents them from activating genes necessary for DNA synthesis. Now Rb could also be hyperphosphorylated, and this could happen due to overactive cyclin D, CDK4, or CDK6. This causes E2F to be released prematurely, and this allows inappropriate S-phase entry. Loss of Rb function, either through mutation or excessive CDK activity, promotes unchecked progression through the cell cycle. Now let's talk about oncogenes. So oncogenes are mutated or overexpressed forms of normal genes that drive cell proliferation. These normal genes are called proto-oncogenes. For example, overexpression of cyclins or CDKs or constitutive activation of growth factor signaling pathways like RAS or HER2 can push the cell past checkpoints regardless of internal conditions. These changes lead to a situation in which cells escape normal growth control, divide indefinitely, and accumulate additional mutations that promote invasion, angiogenesis, and metastasis. This is why understanding cell cycle regulation is essential for studying cancer biology, and why many modern cancer therapies, such as CDK inhibitors, aim to restore cell cycle control. Now we talked about tumor suppressor genes and about p53 and how it's activated in response to DNA damage, and while p53 tries to repair the damage, sometimes the damage is too great or sufficient repair is not possible. In these cases, p53 will cause the cell to die in a process called apoptosis. And so apoptosis, which is also called programmed cell death, is a vital cellular process that works in parallel with the cell cycle to maintain tissue health and homeostasis. When a cell experiences DNA damage that is so great that it cannot be fixed, or when it experiences prolonged cell cycle arrest, or loss of proper signaling, it may undergo apoptosis instead of continuing through the cell cycle. This prevents damaged or abnormal cells from dividing further. Now, one of the key links between the cell cycle and apoptosis is the protein p53. Now, as we've discussed, p53 can pause the cell cycle at the G1S checkpoint by inducing p21. This gives the cell a chance to repair DNA damage. However, if the damage is too severe, p53 can shift its role and initiate apoptosis. This protective mechanism ensures that cells with potential mutations do not continue dividing, and this acts as a final quality control step. Apoptosis can also be triggered in response to mitotic errors or failed checkpoint completion. For example, if the spindle checkpoint is not properly satisfied and chromosomes are missegregated, intrinsic cell stress pathways may activate apoptosis to eliminate the faulty cell. The process itself involves a cascade of caspase enzymes that systematically break down cellular components. The intrinsic pathway of apoptosis is closely tied to mitochondrial health and is regulated by proteins such as BCL2, BAX, and BAC. When activated, this pathway leads to the release of cytochrome C. This triggers the caspase activation and controlled cell dismantling. Now, you probably don't need to know this in too much detail as it's not extremely relevant for the MCAT, but I took a cancer class and I thought it was really interesting how this worked. And so you may recall that cytochrome C is a part of the electron transport chain, and the electron transport chain is in the mitochondria. And so what those proteins do that I talked about, BCL2, BAX, they induce something called MOMP, and MOMP stands for mitochondrial outer membrane permeabilization. This is considered the point of no return, as now caspase C can leak out of the mitochondria, leading to cell death. And so what cancer cells do is they try to stop this process by any means possible. They try to make more receptors on the cell surface that are fake, so you don't get a signal coming into the cell. They try to stop those proteins that trigger membrane permeability. They just want to stay alive. And it's actually really fascinating how cells have so many tricks that they can use to try and stay alive. And if you have a chance, you should definitely take a cancer class or just learn more about it. I think it's really interesting. Okay, so back to apoptosis. By coordinating with cell cycle checkpoints, apoptosis acts as a safeguard against abnormal proliferation. This makes it a crucial counterpart to cell division, ensuring that only healthy cells progress through the cell cycle and divide, while damaged ones are eliminated. Now, apoptosis is not the only fate for a cell under stress. When a cell encounters damage or dysfunction, especially from sources like oxidative stress, it has several possible outcomes depending on the severity and context. Oxidative stress arises when reactive oxygen species, also called ROSs, accumulate beyond the cell's antioxidant capacity. These reactive molecules can damage DNA, proteins, and lipids, posing a major threat to genomic integrity. If ROS-induced damage is detected, the cell may pause the cycle to attempt repair. If the damage is mild and repairable, the cell can resume cycling. If it's too severe, pathways like p53 may trigger apoptosis. I'm going to go on another little tangent here, but they can also trigger ferroptosis, and that is most common in ROS-induced damage. And ferroptosis is essentially a form of cell death, but due to iron. And iron has a big role in ROS accumulation. So obviously, you don't need to know this. I'm just doing my directed studies on this, and so I thought it was kind of interesting. This is what I'm studying, and yeah, I think it's really cool. But moving back, in some cases, cells that accumulate damage but are not eliminated may enter a state known as cellular senescence. Now, senescent cells are metabolically active, but permanently withdrawn from the cell cycle. This means they cannot divide. This is often a response to persistent stress, DNA damage, or telomere shortening, and serves as a long-term protective mechanism to prevent propagation of damaged DNA. However, senescent cells can also secrete inflammatory cytokines and other signaling molecules that affect the tissue environment, a phenomenon known as the senescence-associated secretory phenotype, or SASP. Now, the body's ability to recover from damage depends on regenerative capacity, which is the ability of tissues to replace lost or damaged cells through division. Tissues like skin, GI epithelium, and liver have high regenerative capacity, relying on frequent and well-regulated cell cycle reentry. Other tissues like neurons and cardiac muscle have very limited regenerative potential. These cells are often terminally differentiated and locked in the G0 phase. They are unable to re-enter the cell cycle even after injury. In such tissues, damage accumulates over time, making apoptosis or senescence more consequential. Together, apoptosis, senescence, and regenerative capacity represent three intertwined outcomes of cellular stress and damage. They're all governed by the cell's ability to sense its internal state and regulate progression through cell cycle accordingly. Understanding these responses provide important context for diseases like cancer, aging, and tissue degeneration, where these processes are often disrupted or imbalanced. That's it for today. Next time we'll go over some practice questions.